Thank you everyone and class for inviting me here. My name is Ishay Attar. I'm co-founder and CEO of Exome. Exome is a startup company founded based on patent license from from Shiba in 2014, and we've been developing sustained release systems, products for ophthalmology, for drugs. So far we've been able to develop two products. The first product is for glaucoma, prolonged release prostaglandin analog, latanoprost, and the second product is tacrolimus for treatment of dry eye and allergies. The problem we are really coming to solve here is compliance. Compliance is a major issue when taking medication that come in eye drops. Actually, over time, and this is a glaucoma study, it drops dramatically to less than 30% compliance. So if we want to save people from getting blinded from glaucoma, for instance, because they don't feel anything, and maybe some of them are aged, we got to give them something that gives the drugs, brings the drugs to the eye automatically. And this is where we're at. We're at a sustained release system that gives the existing drug with this known mechanism of action, but gives it over a prolonged period of time. And it's not so easy to achieve engineering-wise a solution that actually does this properly and gives the same results like giving the medications in the eye drops. And there are several issues with it. But once it's achieved, then there's really a lot of advantages besides the compliance issue, which is solved. And also, there's no preservatives, there's no excipients. Only the API itself goes out to the eye. And also, the dose that actually meets the tissue is lower because it's constant. It's released in constant. There's not the bolus effect that you see like in this graph with the ups and downs and big dose and then low dose and variations throughout the day when you give the drugs in eye drops. So our products that we've set out to develop are, first of all, we decided we will go on a non-invasive vehicle, and this is partly because we wanted to differentiate ourselves from the competitors that go into the eye and inject all kind of inserts for sustained release. And the other thing is that we believe that to treat anterior diseases like glaucoma and dry eye, but also others, for instance, we're here in Presbyopia conference, we can also probably in the future treat presbyopia or give some presbyopia APIs that cause meiosis. And doing that with an implant that goes inside is not really for the majority of the patients. They would not want it, and the physicians would not want it. So we need to have some kind of a non-invasive vehicle. We chose to use the punctal plug as the vehicle or as the insert. So we insert a small punctal plug, and that's how we deliver the drug. Of course, because only the API goes out, it's preservative-free. Everything is packed inside the vehicle. The problem is retention. Retention is never 100% when it's non-invasive. It's around 90%. So how did we achieve it technologically? What do we do? So that's our core know-how and technology. We use certain materials that have a huge surface area. That means that if you take like 1 gram of this material that we use to pack the API and unfold that 1 gram, you get like 200 meters square. It's like a basketball court. And then we use solvents to load the API. We pack it back, we evaporate solvent, then we get another powder that contains a lot of API, and then we make it a physical shape using epoxy, a glue, and some other another substance, some kind of a bulking agent. And that's where we get certain composite that we can use to shape it into any shape we want, because it takes it like 24 hours to harden, to set. And it contains everything, contains the drug particles in it. And the drug, and the interesting part of it is that they don't all go out at once. So we have like a composite that's about 30% weight-to-weight loaded with the API, but it doesn't just go. And there's no barrier. The barrier is the composite itself, or the nanopores in the barrier that disturb, or they do not let all the API go out at once. The API wants to go out only when it sees water because of diffusion, and they can't. It's like a traffic jam out of a big city. Everyone would just want to go out, but just takes time and they go out slowly. We can take this composite initially and shape it into any form we want. We use standard molding procedures to mold this into any shape. We mold it into the punctal plug. Punctal plugs are known, they're being used for treating dry eye, the passive plug from silicone, so we copy paste the same shapes. We're not getting into like development of new shapes and things like that. We just have a plug that release the drug for a prolonged period of time. And also the scale up of it is very easy to do. Punctal plugs have been used a lot. There's a lot of data in the literature about the comfort level of punctal plugs, retention, and so it's not perfect, but it's the best solution that we could put our hands on, and that's where we decided to go on punctal plugs. So we're going after large markets of glaucoma and dry eye and allergies right now, which there's no product in the market that gives a sustained release solution. Not yet. There are a few others that develop. Our first product is contained in a prolonged release latanoprose for glaucoma. We can also load two drugs on one plug. So we can have both of them release simultaneously from one plug. And we tried this with a latanoprose and a timolol. Although there are two different molecules, we got both of them to release at once. And that's important like for the next phase of next products, because half of the glaucoma population, they're on dual therapy or triple therapy. So they need more than one drug. This is an example of a release curve of what it looks like. Initially, we give a kind of a boost, more dose for the first 30 days, and then it stabilizes to a zero, the release curve over time, and continues with it like for 6 months. We tried this first in the normotensive beagle dog model, and actually it worked very well. And I think like also relevant for this conference, the presbyopia, you can see the meiosis in the dog caused by the latanoprose. That's a side effect of latanoprose, which is only relevant to beagles, to canines, not to humans. But in humans, we can achieve this with other pilocarpine, for instance, and meiosis can help with the presbyopia, obviously. We've also measured, of course, the effect on the IOP, because that's the goal of this product was to reduce IOP, and the normotensive glaucoma reduced IOP by 50%, which is comparable to the drug therapy, the xalatan, the standard of care for glaucoma treatment, or the same drug with the same API but given in the eye drops. Our next phase is starting October, is a clinical study. First in men, we're going to do this in Shaarei Tzedek hospital department. 3 months follow-up after 1 month washout. And we are looking for and eager to start this study and start generating human data. Next product I'll talk about is the tacrolimus. So the problem of dry eye, you all know, is huge, and also with the allergies. About 7% of all the population is affected by it. Of course, some of it is mild, but there's also a lot of severe cases, and especially in Asia. The current drug, which is the oldest drug and the main drug, is cyclosporine, given in Restasis, given in the eye drop, blockbuster of $1.5 billion a year. We just fell off the patent for Allergan, and people are looking for new solutions in this market. So we came up with the tacrolimus. Tacrolimus is very similar to the mechanism of action to the cyclosporine. It's an immunomodulatory agent, just like the cyclosporine, but it's less hydrophobic, so it's easier to get it out of the plug and to the eye. And it's about 50 to 100 times more effective than cyclosporine. So it's actually seems like a perfect molecule. It's not approved for dry eye right now, but it's been studied. People are taking this as self-formulated it and gave it and did some studies, even 48 eyes, one study, 16 eyes, another study, and Canaan studies, and it works very, very well on the dry eye syndrome. It is approved in China and Japan for allergies. We can also demonstrate and show how we can do a sustained release curve of tacrolimus over a prolonged period of time. It's about 1.5 micrograms a day, which is released, which is lower than the dose that's given in the eye drop. But there's really a very vague correlation between the dosing of the eye drop, the medication given in eye drop, to the dosing of the medication in a sustained release system. It's the physiology, the eye reacts very differently to a sustained release comparing to a regular bolus eye drop. So it's, we got to do it empirically and find the right dose. So we tried this first on dogs that came from the community and they were sick with KCS. They had actually, first patient had 0 tears, almost blind, no tears, very severe case. The normal reference for those kind of dogs is about 15 to 25 millimeters a minute. And you can see at baseline, and then after 14 days, 42 days, up to 105 days, very encouraging, but very initial results that we need to go forward with this product. We are now working our way to the clinical studies with both products. We also will be interested in the future to develop other products as well for any front of the eye APIs that have an effect that may open up new market. Presbyopia market could be one of those. Also the myopia story with the atropine could be interesting. And that's it. Thank you. If you have any questions,